Okay, so good morning, everyone. I'm still puzzled that we have only 5 people at the moment. I like virology a little more than bacteriology, so if anything, I'm a trained virologist. So, and this allows me to go at times beyond the textbook and make it a bit more interesting, I hope at least. So today we have 2 lectures. The first one, I will give an introduction into virology and maybe a mini introduction into immune responses as well. And then we have a big topic, and that is HIV/AIDS. And HIV/AIDS has been moved a little bit in the background when the whole corona pandemic started, but I think this is nothing that should be neglected, and there are still many, many unresolved issues. Okay, so you will find stuff on viruses in many different places in the textbook. Here are the main chapters, but I took an approach that is, I picked some virus families and some viruses that are what I think of imminent medical importance, and so it's a bit of a personal choice. Okay, let's start with the fact that viruses are obligate parasites, and many people do not regard them as alive, but as we will see later, the picture blurs a little bit, and this is a bit of a philosophical question, so it's not really worth dwelling there. So the characteristic features are that viruses multiply inside living cells. So it's like a computer virus needs a computer to multiply, and viruses need a cell to multiply. However, as far as the cell goes, they're rather promiscuous. So bacteria, protozoa, fungi, plants, animals, all this goes. So unfortunately, for all those who study viruses, what consists a genome, a viral genome, is not that well defined. Well, no, it's defined, but it's flexible. It could be DNA, it could be RNA, and as we will see, also weird intermediate forms. So infected host cells produce new viral particles that transfer the viral genome to other cells. So that's the whole purpose of a virus. It's a vehicle to get from one host cell to the next. The viral genome in a virus particle is usually protected by a protein coat that sometimes itself is enclosed by an envelope of lipids containing further proteins and carbohydrates. So sometimes people think, oh, why should I learn whether this is an envelope virus or not? Because if it's an envelope virus, you can already tell a little bit about how it can infect cells and also how you can kill it. If a virus has an envelope, it is usually easier to kill than without an envelope. So viruses may have more than 400 genes, but nevertheless, they cannot generate proteins and metabolic energy on their own. That's why they depend on living cells. So I showed this photo before because it contains a lot of things we are concerned with in mechanisms of disease. For reference, in the right corner here we have the extent of a human red blood cell, and here we have bacteria and viruses. So there are a few things crossed out, and viroids are sometimes not classified as viruses, but I wouldn't do this, but you know, this is a textbook, because it contains RNA only, and the RNA is not protected by a protein coat. Then prions are definitely not viruses, because prions are, by the end of the day, cellular proteins that can propagate, but not because they make new proteins. What propagates in the case of prions is a wrong conformation of the protein. And I will have like an entire lecture on this. So there were 2 Nobel Prizes awarded to resolve the mysteries associated with prion diseases. So it's pretty important. It breaks all kinds of dogmas, and is very interesting. And then chlamydia, the last thing that's crossed out here, they are small intercellular bacteria, probably related to cyanobacteria, and the genome is much reduced. So, and they are so small, they are often dealt with in all the textbooks under viruses, but they're actually bacteria, not viruses. Okay, so the following is a little bit for your amusement only, but I'm not going to assess you on it. So there is a committee on the taxonomy of viruses, and that kind of governs how these things are named, and who can name what, and what names are appropriate and what aren't. And this is all to make searching and dealing with viruses easier, because previously there were several viruses named so similar that they had the same abbreviation, and that would just mess up everything. So viruses are classified by the type of genome they have, the nucleic acid type, DNA or RNA, whether it's fragmented, and its organization, and in the end also its sequence. By their strategy of replication, and replication here means genome replication, and they morphology, so for example, whether or not they have an envelope on top of the protein code. So to understand this, however, is a little easier, because all viruses in a virus family behave quite similar. So if you know one herpes virus, you can deduce a lot for the other herpes viruses. So, and if it comes to naming, so what we have here at the bottom is a species, and the next level on top of that is a genus, subfamily, family, order. This is pretty much similar to zoology. And these are the different endings. So if, for example, if I say Paramexoviridae, then I speak of a family. And measles virus is a species. So what is kind of important in a way that makes you look competent if you speak of viruses and the diseases they cause? These things are not necessarily the same. So the measles virus causes a disease that's called measles. But if you just say measles, then you speak of the disease rather than the virus. So you may think it's splitting hairs, but sometimes this is actually important. Okay, and I told you that viruses can infect all kinds of things. So for example, bacteria, Eubacteria, and Archaea. But we will not really go there because obviously not of medical importance. Then viruses infect vertebrates, and we are one of them. So that is then much more important. And here we have the one that caused such a stir a few years ago, and that is the SARS coronavirus 2, and that belongs to the family Coronaviridae. And that is a single-stranded RNA virus that has a genome of positive orientation. I will come to this in a minute. And that then belongs to the RNA virus group. So there are also viruses that infect plants. They are important because they can cause dramatic losses, but we will not deal with it in this lecture. So, and then there is also something that we will not deal with, but I like to mention it because I think it's kind of funny. So this is a front cover from the year 2011, and what is kind of a clumsy modeling of very large viruses. And what we see here in front is the size of an HIV particle, and what we see in the back is the size of so-called mimiviruses, and that stands for mimicking microbes, but a virus, so not able to do protein synthesis of its own, still relying on cells to multiply. These viruses are the largest, together with the megavirus and the Pandora virus, the largest viruses found so far. And they were discovered last, not early, which is a bit weird that the biggest thing, took longer to discover. Okay, so they're pushing the boat out in some aspects of of conventional definition of a virus, but it's still holding true that all of those absolutely depend on living cells for multiplication. Okay, now I want to come back to RNA and DNA, and this is now eminently important because that's subject matter to assessment. So this is just for convenience. You should know what an RNA is and what a DNA. So it's not the same thing. So it sits different in different places when it comes to the storing or transferring of information. So we, us, store most of our genetic information in the form of DNA, and then we transcribe it into RNA, and then we translate it into protein, and then we have action. So that's the general dogma. So viruses can, however, store the information permanently in RNA, and they don't use DNA at all. So therefore, it might be good to actually know the difference between RNA and DNA. And in general terms, one difference is that RNA has one oxygen molecule in the backbone more than DNA, so one hydroxy group here where there is only an hydrogen atom here, and that makes RNA more reactive. So DNA, if you like, is the safer and more stable storage form. So for us it's good because we want to keep most of the time our information very stable, and we do not like mutations because they're most of the time deleterious. Whereas viruses operate in much higher numbers, they can afford all this, and for some it is quite effective if they just don't use DNA, as we will see. So whether a virus uses DNA or RNA will not tell you whether it's more or less dangerous. So here is a little principal stuff on RNA viruses. They are basically sub-classified depending on whether the genome contains double-stranded RNA or single-stranded RNA. And the information flow for viruses with double-stranded RNA is from here, they copy it into messenger RNA, and then they can translate it into proteins. If you have single-stranded RNA as the genome, and the RNA is of positive orientation, the definition for this is positive RNA equals mRNA. So it can be translated straight away. Negative RNA is basically a copy of positive RNA. To make mRNA of this, you have to copy it first into positive, or dedicated positive RNA, like mRNA, and then you can translate it. So you see that these ones, once they infected a cell, it's only one step away from making more protein. These are 2 steps away from making more protein. So you can see here where this is going for some of the viruses. So they basically make it super efficient. They do not care about whether it has high fidelity or not. They want to be quick. Okay, now we come to DNA viruses and the double-stranded DNA viruses, and the example here, sorry, I do have example reoviruses here, polio hepatitis virus here, and influenza measles virus here. So we will talk about hepatitis C, and we will talk about influenza in subsequent lectures. The herpes and the pox viruses, they're a bit like us. They have double-stranded DNA as a genome. Then, if they want to make proteins, they have to transcribe this into mRNA, and then they translate it. The single-stranded DNA viruses are a little bit weird because they first complete the DNA to double-stranded DNA, and then they do the same as the double-stranded DNA viruses. So this is basically quite elaborate here. And then we have reverse transcribing viruses. These are truly weird. However, you need to learn it because in the second lecture today, we will talk about HIV/AIDS, and that is a reverse transcribing virus. So the stuff that is in the virus particle, that's always on top of this list, so that defines what a virus is single-stranded RNA. So every HIV particle inside has 2 identical copies of single-stranded RNA. That doesn't make it double-stranded. It's 2 copies of single-stranded RNA. And if that gets into a suitable host cell, then the RNA is reverse transcribed into DNA, a highly unusual step we rarely do. Does anyone know where we do this? There is a notable exception to the rule. 5 brownie points. There was a Nobel Prize given to this, and I think the person worked for a year in Tasmania, for a while in Tasmania. Hmm? No. You do it as we speak. Yeah?
What was the question again?
The question is, so these viruses, the reverse transcribing viruses, they have a critical step here, where the RNA is reverse transcribed into DNA. So all your messenger RNAs are properly transcribed. So the information flows from DNA to RNA. So that's what you learned in school, basically. So here these viruses have one step where they go from RNA to DNA. But I tell you now that you and I, and we all, do this every now and then as well in our cells. Does anyone know what the purpose of that is? Do you know the ends of your chromosome, the name for it? Telomeres. Telomeres. Yeah, and there is a step to maintain the telomere lengths, or at least to attempt to maintain the telomere lengths, that involves reverse transcription. So that's, to my knowledge, the only time we do it in our cells. So it's extremely rare, which is good. It is one thing that's associated with aging. So, okay, but this is good because we do not do this step. That makes these viruses extremely vulnerable if you want to develop antiviral drugs, because their replication machinery, their polymerase, is different from all our polymerases because it has to do this step. And that makes them vulnerable for inhibition, and then you get fewer side effects. Okay, good. So once they transcribe the RNA into DNA, and then they do the single-stranded DNA into double-stranded DNA, and then they integrate it into our genome, into the genome of the host cell. And then they make mRNA, and then they make the protein. So you see, this is horribly complicated, but I will spend, the next hour, a portion of the next hour, on giving you a concrete example for this. And you need to learn it because there's no doubt that HIV/AIDS is of imminent importance and will remain so for some time.
Just one last question about that reverse transcription from RNA to DNA. Does that have to do with, does that prevent the virus from aging?
No. The virus is not really concerned about this. Yep. Okay, so now the virus morphology. So there are several morphological types, and they are based on the capsid structure. We have polyhedral viruses and enveloped polyhedral viruses. And here we have two examples of a non-enveloped and an enveloped polyhedral virus. It looks a little fluffy and normally is a little bit more structured and not as fluffy, but this has been destroyed to show more easily that there is basically a protein shell inside that envelope. That is, I'm tracing here now with the laser pointer. And inside that protein shell, we can see here is the genome, which is either RNA or DNA. So then we have helical viruses and enveloped helical viruses. So that means that the capsid proteins that make that shell are a little differently organized, and that gives us these viruses. So on top we have a plant virus, and here we have the measles virus. Okay, another thing is that if you have an enveloped, you often have proteins, particular proteins, sitting in that envelope, and these are sometimes referred to as spikes, and they can be imaged. So what we see here are influenza viruses, and you see the spikes kind of surrounding the virus particle. And then we have very fancy viruses that look a little bit like starships. So these are called complex viruses. We will talk a little bit about this Gesundheit now, but not much about it later, because these viruses infect bacteria. And however, we can use it to show the principle of multiplication. So here we have several steps in the multiplication cycle. The first step, and that's true for most viruses, is an attachment. So the virus has to pick the host cell, and that can be more or less specific. So then in this case, because it's a bacterial cell and a bacteriophage, the virus actually has to drill through the cell membrane and through the cell wall. And the cell wall is rather rigid. That's why we have all this machinery here, and you know now all about the cell wall. And then it injects its genome, and then the genome is transcribed, replicated, and translated. And if it's translated, you end up with a lot of viral proteins, and they often have the tendency to self-assemble. And then you get, in the end, daughter viruses or progeny viruses, and they are released more often than not by the destruction of the host cell. And there are different strategies, as we will see later. Some viruses just go hell for leather. They don't care whether the host cell survives or not. They use up all the resources. They go for speed, and they make as many virus particles as possible in the shortest period of time. The other strategy is to hide in an infected cell, to release fewer virus particles, not to kill the infected cell, but then to release these viral particles for a much longer period of time. Both strategies can be rather successful, but however, if you have the strategy like a sustained use of the host cell, then you run into problems as a virus. If you do that with organisms such as us, because we have then time to mount an immune response, and you have to deal with this. And then it gets really complicated, and we see a few examples later. Okay, bacteriophages, just for those who want to use them later as tools, they can actually form plaques. What we see here is what you know from the practicals. So this is a bacterial plate that has been swapped, and there are bacteria everywhere. But then if you seed viruses onto those bacteria, and these viruses are lytic, then they can eat, if you like, holes in the lawn of the bacteria, and you can see them here. They're called plaques. And you can use that to determine the concentration of a suspension of bacteriophages by diluting the suspension and inoculating samples onto your bacterial plates. And here we have tenfold dilutions, and you see that with every step you dilute them, the number of plaques is also reduced. Okay, that allows me to show the difference between the lytic and the lysogenic cycle. So what we see here to the left, I just explored a few slides ago. What we see here to the right is an integration of the viral genome, that is that little red bit here, into the bacterial genome, and then the virus doesn't need to do anything, because the information will be doubled every single time the bacterial cell doubles its genome and divides. So it will be propagated with the bacteria. But as with many bacteriophages, the viruses under certain conditions may decide to kill the host and to go for daughter viruses that then can infect other hosts horizontally and not just spreading through the generations of bacteria. Okay, so here we have the multiplication of a DNA virus as an example. So this is just a generic virus, and we see a virus particle with the genome inside, and the steps are very similar. So we have an attachment step, and this step can be fairly specific, and as we will see later with HIV, it's very important to know this step because we can then make antivirals to stop this from happening. So in this case, contact has been made, and then the virus has to find a way to move the genome into the cytoplasm of the host cell, and that is then what we call infection. So the virus enters the cell, DNA, in this case it's DNA, it's uncoated, and then in this case the virus enters the nucleus, so it's moved from the cytoplasm into the nucleus, mRNA is made, the mRNA then needs to leave the nucleus, then it is translated, you make new virus particles, they spontaneously assemble, they grab a new copy of the genome, which also replicates here while you make new proteins, and then you get the formation of new virus particles, and then there has a mechanism to release them. So either by disrupting the cell and just releasing everything at once, or like a more sustainable way where you have exocytosis so you do not damage the host cell. Okay, here we have a detailed view, a generic view, nevertheless, of the virus entry. So here we have the entry of an envelope virus by fusing with the plasma membrane. So here we have the virus particle, we have the envelope here in blue, we have the capsid in orange, and we have the nucleic acid, the genome in red. So, and then we have our spike proteins, and the spike proteins make contact with the receptor. That is usually one of the proteins on host cells, and that can give the virus a certain specificity in terms of HIV. The main receptor is CD4, so that means HIV only infects CD4 positive cells. So if a cell doesn't have a CD4 protein on its surface, it is safe from HIV. Okay, so here successful contact has been made, and this kind of works in a funny way that if you put the membranes of the virus particle and the membrane of the cell close enough, then there is a tendency that they fuse. And if they do, then you provide access for the capsid, so the whole thing can then enter the cytoplasm. And then the virus is inside the cell. Here we have an entry of envelope viruses by endocytosis. So this is one additional layer of complication, but similar mechanism. So you have the virus particle binding a receptor, but this then triggers the formation of a vesicle, endocytosis, by the host cell. So the host cell basically, if you like, don't write it in assessment, eats the virus particle. So then the virus particle is inside the host cell, but not yet inside the cytoplasm because it sits within that vesicle. And then another principle is that these vesicles that are generated by the host cell, they tend to have the pH dropping in their in the lumen, that is in the enclosed volume. So protons are pumped in, and the pH in an endosome drops. This is usually done because the cell triggers this to eat up things it has just taken up. So, and that is favored with low pH. So the dropping of the internal lumen of the endosome causes conformational changes in the virus proteins, and that then brings the membranes, that's not really shown properly here, so close together that you get the fusion event now and not at the very beginning here. And that then leads the capsid leaving the endosome. So this is a step, for example, that is taken by influenza viruses. And there used to be a fairly efficient drug that stops it, but it has been overused in pigs, and now it doesn't work anymore. So, so it is important to study these things because it opens possibilities for antiviral invention.
Does the coated vesicle become the endosome? The previous
Yes, this is the first step is called invagination, and then we have a cultured vesicle, and that is then an endosome once it has detached itself from the plasma membrane. And then it goes a certain path that involves dropping pH and so on. Okay, then we have entry of non-enveloped viruses by endocytosis, so very similar, but no fusion of membranes because these viruses don't have any. And the opposite is if a virus wants to leave a host cell, then if it's an enveloped virus, then it has to acquire the envelope from somewhere. And depending on how it does it, the envelope could come from the plasma membrane, the nuclear membrane, the ER, or the Golgi. And the process is shown here. But the process is very similar to regardless where the membrane is basically stolen from. Okay, and this is an important slide because it shows you what can happen if a virus infects a host cell. I kind of referred to this before, but this is now like a nicer graphic depiction. So an acute infection is if a virus infects a host cell, then we have the multiplication of the virus inside the host cell, and the host cell is destroyed and all the viruses are released. That's called an acute infection. A latent infection is the viruses infect the host cell, or a virus infects a host cell. Viral components are made, but the host cell is not killed. So that's called a latent infection. But the latent infection will not lead to the spread of new virus particles. So there could be an activation, and then it is completed, the host cell is killed, viruses are released. Then we have a chronic infection. We have virus infecting a host cell, being a little bit more careful with the resources, a constant release of new virus particles, perhaps not as many as in an acute infection, but the host cell survives. And then we have basically an accident, and that is if the virus infection leads to cancer. So this is not really helping the virus, but the fact that many viruses integrate into the host cell genome can lead to unintended consequences. So that is basically messing around with our genes. And this is an opportunity for malignant transformation, and that is a cell becoming a cancer cell. And then you have the spread of these cells. By that you can also spread virus information within the cancer cells, but often this does not lead to the production of new virus particles. So from a virus point of view, this can be considered an accident. And if the cancer kills the host, it's also not really helping the virus. So, but nevertheless, it happens. And depending on what virus we are talking about, it can happen quite frequently. So hepatitis B and hepatitis C are often associated with the development of liver cancer. That doesn't really help the spread of the virus, but nevertheless kills the host. Any questions to this? Okay, so now we have a bit of time to give a kind of a bit of a crash course in immunity. So what I liked, and I will repeat this when we talk about immunology later. So you can look at immunity in kind of the time it takes to get certain responses. And then you can divide immunity into innate immunity and adaptive immunity. And innate immunity is, you can say, it's the first and second line of defense. So it is very fast, so within hours, whereas the adaptive immunity takes time, is very powerful, often gets rid of an infection, but takes days, if not weeks, to mount. So, and then people say, oh, who cares about innate immunity? Adaptive immunity is basically what kills an infection. If you do not make a powerful adaptive immunity and do not control a virus, then it often kills you. For example, HIV. People do not manage to produce a powerful adaptive immunity, and then earlier or later they die as a consequence of the infection, which has led for a while to a neglect of the innate immunity. But what people forget is if you do not mount an innate immunity, you never survive long enough to mount an adaptive immunity. So the innate immunity buys you time to do the adaptive immunity thing, which takes 2-3 weeks to become fully effective. So an important part of the innate immunity are interferons, and interferon is a particular class of cytokine, and these are messenger molecules, little proteins, and they are released by virus-infected cells to warn other cells that aren't infected yet that a virus is about. And then these cells can prepare themselves, and if that works well, then the virus cannot replicate efficiently or cannot replicate at all in cells that have been warned. And then you may think, oh, why not warning all the cells all the time? But this is a little bit a costly thing to do so. So we do this only if there is a threat. So this is how it works. So this is supposed to be a cell, and this little blob here is supposed to be the nucleus. And then we have a virus, and you know by now that the virus tries to enter a suitable host cell, then it multiplies, and we get several viruses coming out of this. But the host cell is not completely defenseless. This host cell has several ways to recognize that it has been infected. So for example, there is too much RNA about, or there is RNA of the wrong type about, or there's all kinds of patterns associated with a virus infection. So this cell can become aware of the infection, and if it does, then it transcribes proteins, that transcribes genes and makes proteins it normally does not make. And one class of those are the interferons, and these are the messengers that are released, and then it's a little bit of a race to get your newly produced viruses and to get interferon. And if you get interferon made first, then the interferon can warn neighboring cells, and they make antiviral proteins, and if a virus tries to infect such a cell, that's a dead end for the virus. Yes?
Interferon is also a medication used to treat hepatitis C, I believe.
It used to be a medication to treat hepatitis C. Yes, with varying success depending on the genotype and lots of side effects. But these days, almost all people are treated with specific antivirals, not with interferon anymore. And that clears the infection. Okay, now we come to the adaptive immunity. And the adaptive immunity, there are two types of cells very important. It's the T cells and the B cells. The T cells, they are there to kill virus-infected cells. They also can make interferons and other cytokines, but they kill the viruses by killing the cells with the viruses. So, and you can imagine that this can lead to some side effects. So for example, if you have a liver infection and then you have a vigorous T cell response and you kill 20-30% of all your liver cells, you will be sick. You may get rid of the infection, but if you overshoot, this process can even kill you. So, and then we have B cells. The B cells, they do not directly kill virus-infected cells, but they make antibodies, and the antibodies can then bind to virus particles outside of their host cells and then make a coat around them and neutralize them, and then if they are coated in that way, then they struggle to get into new host cells. So the antibodies are some sort of interceptors, if you like. So this is a bit shown here. So B cells are cells with the ability to produce antibodies, and we will get to this in great detail at the end of the lecture series. But these B cells, if they get activated, they can transform themselves or differentiate into plasma cells, and these are kind of factories for antibodies, and they produce large amount of antibodies. What you see here looks like a Y. This is an IgG type antibody, and what you see here, bacteria, and these antibodies can bind to bacteria and then inactivate them. So the T cells, they can kill virus-infected cells. That is shown here to the right with the cells in pink. So if they get activated, then they go after cells that they detect have viruses in them. And this is possible because most virus-infected cells show little bits and pieces of the virus proteins on their outside. And these T cells just scan all your body cells, and if they find something they don't know, they just assume it shouldn't be there, and then they kill the cell. So it's basically they leave the cells alone if they don't find anything new. And they are basically, they don't ask too many questions. So if they find something that they haven't seen before, they kill that cell. And the blue cells here are also T cells, and they do not directly kill virus-infected cells like the cytotoxic T cells do. But there is an important population, the T helper cells. And the T helper cells, they help through cytokine secretion the cytotoxic T cells to do their job, and they also help the B cells to differentiate, become plasma cells, and produce antibodies. So if you like, even if the T cells themselves do not seem to kill viruses directly, they are absolutely critical for both arms to do their job. So as we see with HIV, because HIV targets the T helper cells, and that really wrecks our adaptive immune response. And that's why people who got infected with HIV normally, up to now, stay infected until they die, either of HIV or of other causes, but they will not get rid of the infection. Okay, and I told you that an adaptive immune response takes time. And this slide, which I will show again, kind of explains why this is the case. Here, it starts with a stem cell in your bone marrow. And this stem cell does something unusual because it is allowed to make gene rearrangements. It actually tosses things out of your genome. They're lost forever. But these cells will not become ever, egg cell and sperm cell. So they're allowed to do this. This is an exception from the rule. And then they rearrange the genes, for example, in this case, for producing antibodies. And then they can produce, through that rearrangement and other tricks, an almost unimaginable variation of antibodies at random. It's a completely wasteful process. So they end up with a lot of cells that produce different antibodies, not knowing which antibody will eventually be used. And then most of these antibodies are for the bin. They will never be used at all. These cells are made and die without having ever done anything meaningful. But if one of these cells clicks with the pathogen, here we see a little virus, then this cell gets really excited, and this cell then gets the permission to multiply. And then this single cell that was generated at random, and all the other thousands combinations were for the bin, this one hit the lottery ticket and was allowed to become a clone. And it multiplies, and then all of these cells produce the same antibody that was picked in this initial process. And then you make tons of that antibody against something new that has been encountered by that cell. And you can imagine there is a lot of differentiation, multiplication, and growing up of cell numbers, and that takes 2 to 3 weeks to become fully effective. And that's why the adaptive immune system takes time. Okay. In the upcoming lectures, I think we're pretty much on time for the first lecture. You will learn about the human immunodeficiency virus. That's one we do after a 5-minute break, and that's the one that famously transcribes its RNA genome into DNA, and then it integrates into chromosomes, and it wrecks your immune system, and if left untreated, has a high chance of killing you. Then we will talk about herpes viruses. You may be interested in herpes virus because nearly, if not all of you, are infected with herpes viruses, and this will stay that way. However, there are some herpes viruses where the infection can now be prevented by vaccination, and that is, for example, varicella zoster. That is the disease that can be stopped by stopping people to get chickenpox in the first place. So we will talk about this in the herpes virus lecture. Then I have one lecture on hepatitis viruses. This is not a family. So herpes viruses are a family, so you can say all herpes viruses are quite similar. But the hepatitis viruses, they have basically nothing in common. The only thing they have in common is that they go after your liver. But the viruses themselves are quite different. So we have RNA viruses, and DNA viruses, and even retroviruses. So that makes them all different. And if you know one, you do not necessarily know anything about the other. Whereas if you know something about one herpes virus, you know already a lot about the next herpes virus. Okay, but because hepatitis is still important, so there are still millions of people running around with hepatitis C and B, and it will cost lots of money to deal with the consequences, I think it makes a good topic. Then I will talk about influenza viruses, because that used to be the real killer. So then came coronaviruses, and now people think, influenza is not important. But the next big pandemic may well be influenza viruses, and that could well be more dramatic than the last coronavirus pandemic. So if you think coronavirus was bad, then you really do not want to have a highly pathogenic influenza virus spreading. So therefore, I think as a virologist, you should never discard influenza viruses, plus they are fairly weird for RNA viruses, which is what I like.
I heard a dose of influenza A that was worse than coronavirus. It was worse physical feeling and like sicker for longer.
Yeah, that depends a little bit. If you ask the people who are seriously sick with coronaviruses and die, they may differ, but yes, on average, influenza virus can kill perhaps more people than coronaviruses. Plus, influenza virus has a propensity also to kill young people. And the coronaviruses, or at least the ones that spread so far, the older you get, the worse it is. But this might be different with influenza. Okay, and then we will talk on coronaviruses. So this is a fairly new lecture for obvious reasons. And then I may also talk about caliciviruses. And caliciviruses is a very Australian topic, because that's what we use to control the rabbits in this country. And I work every now and then with caliciviruses in the rabbit control team. Okay, these are the learning objectives. That was the first lecture. And 5 minutes, I switch lights on, you have a walk around, and then we do HIV/AIDS. And HIV/AIDS has always been assessed. So this is, for those at home as well, another hint. Okay. But you can still ask questions. It gives me something to do in the 5 minutes. if you eat a rabbit and the rabbit is infected with either calicivirus or myxomatosis virus, it's not going to do anything to you. I wouldn't recommend it, but it's not. This is so species specific. There's no way you attract the virus.
I think so. I remember the whole community was like, oh, we can't eat them anymore, we can't eat them anymore.
No, these days, plus the time a rabbit is actually infected with calicivirus is very short. It's usually very short. So the majority of the virus, the rabbits that die within 2 or 3 days. So the chance you're shooting one that is infected with calicivirus is low. It's very low.
My dad about this because we were affected a whole bunch of myths when we were kids, like back in like the 90s and stuff like that.
No, this is slightly different. If it's the early 90s, before the release of the calicivirus, the myxoma virus is different because that takes its sweet time to kill the rabbit. So yes, you have higher chances to shoot it, but we do a lot of testing and people can send their rabbit samples to us and we test. But we don't want rabbits that are shot for the reason that we hardly ever find calicivirus in them. So if you have a dead rabbit in your paddock that died not because you shot it, then you have a much higher chance it was killed by a virus. You're just killing the next 10 rabbits, they are possibly likely all negative.
Like little baby rabbits, pretty big problem, the kittens or whatever, they only get infected after a certain amount of weeks or something?
Yes, the thing is a little complex in that the original virus that was released could not kill kittens up to 3 weeks of age. So when they transitioned from drinking milk to eating vegetable matter, then they became susceptible and then the virus was able to kill them. However, the virus that's circulating at the moment has a propensity to kill young and old rabbits. So yeah, which is in many ways an improvement.
Very interesting because when I saw the police stuff and a bunch of people just went back to childhood and remember all the farms and everything talking about it was a big thing.
Yeah, I think we need a next generation of calicivirus biocontrol agents because there is a problem with rabbits being exposed at an unfortunate time, so they have the chance to survive, and then they will actually acquire immunity, and then as long as they live, they're not likely to be killed by caliciviruses, and then they can be the seeders for the next generation of rabbits. So it would be good to have viruses that do not cross-react to existing antibodies, so and that requires new biocontrol agents. So that will, let's hope our government sees it the same way, and that will then give us more money to develop that. So that's not available yet. Okay, are we complete again? Okay, then we continue. I get the lights off. So this is not the first proper lecture on viruses. So it's not like generic anymore. So we're now really talking about a particular one. And we talk about the human immunodeficiency virus, or abbreviated, let me switch this on, abbreviated HIV. So you see, this is the virus or the etiologic agent, so the thing that makes you sick, and this is a disease, or it's rather a syndrome, the acquired immune deficiency syndrome, AIDS. So this thing here, AIDS, is caused by this thing here, which happens to be a virus. So two different things. Okay, HIV AIDS worldwide. So we see that we have still millions of people, particularly in Southeast Asia, in Sub-Saharan Africa, but also in the Americas, including North America, that are infected with HIV. And this is really bad because if left untreated, the infection is likely to kill these people. However, you can now treat HIV, which gives these people a much longer life, maybe even, judgment is kind of still out, an almost normal life as far as life expectancy goes. And the treatment also reduces the chance that these people infect people that aren't infected yet. However, you have to treat these people as long as they live, which you can imagine, A, costs a lot of money. I mean, this is like the dream for the pharma industry. To have a problem, they have to sell pills for the rest of the life of these people. Plus, then if for whatever reason people decide not to be treated anymore, then the virus has again a high chance of spreading to other people, which doesn't make the problem any smaller. So there are now 39 million people living with HIV, obviously was the last numbers, so the numbers always lack a little bit. And the numbers were actually stable in recent years. So there were about 1.3 million people that became infected per year, and there are about 500,000 people that die every year with AIDS-related illnesses, and this can be a horrible death. And there are 29 million people who receive antiviral therapy. So 29 out of 39. So that means 10 million do not receive antiviral therapy, and that is the reason why the virus spreads so much. So, and you can also tell that whenever you have major disruptions, so wars, people moving from one country to the other, so this thing stops being effectively applied. So then these people are not treated, and then it spreads. Okay, so HIV prevalence among adults, it's, the more red it gets, the higher the numbers. You see it concentrates in some areas of the world, and it can be more than 20%. And this is an economic burden we cannot imagine here in Australia. So, and that is really bad, and that needs to be addressed, otherwise the problem will not go away, and it will continue to spread then out of Africa to the rest of the world, because you cannot contain viruses, as we have learned with the pandemic. So, so Australia also has HIV, and it's also spreading in Australia, even if it's not spreading as much as it is in other parts of the world. And the first recorded case in Australia was in Sydney in October 1982, as far as I know. And the first Australian death was in July 83. So we, in general, in Australia, did a very good job very early on. So there was an aggressive campaign to inform people on how it spreads, what can be done about it, and also later on to take up medication and to pay for the medication. So therefore our problem is not as big as in other parts of the world. So therefore information is key, and this is why I try to have this particular virus as the opener of the specific viruses we will talk about in the next couple of weeks. So how the virus is transmitted is by unprotected sex, kissing, petting, safe, by sharing needles and syringes, very efficient way of transmitting the virus, by blood or blood products. However, big however here, since 1985, the Australian blood supply has been screened for HIV, which has greatly reduced the risk of getting infected by having an accident and getting a blood donation. So it's never zero, but it's close to zero. So previously, babies from HIV-infected mothers had a high chance of becoming HIV-infected straight away. But this has been dramatically reduced by all kinds of measures. For example, cesarean, stopping breastfeeding, and also most importantly perhaps, by putting the mother under antiviral therapy that reduces the viral load in the mother to a point where she is barely infectious. Okay, if we come to the discovery of HIV/AIDS, and credit goes to the CDC, which once was a truly independent, excellent institution. I'm not sure how much of this has survived, but they should be given the credit for discovering this drama. And they found out that young men in California came down with an otherwise super rare infection. So pneumocystis carinii causes pneumonia, is a fungus, and normally as a doctor you never see a case. Super rare. And they had several of these cases around one city. And that is kind of rang all the alarm bells why these people are dying. And then these cases also showed cytomegalovirus-caused disease, signs and symptoms, and they had candida infections. And then people got a little bit the idea that these people with that rare pneumonia have a suppression of the immune system. So something reduces their ability to mount an adaptive immune response. And then they get all kinds of diseases people normally don't get, although the pathogens are around. So many people are infected with the cytomegalovirus, and it never will do anything to them, make them barely sick. But if your immune system is severely suppressed, then the cytomegalovirus can kill you. So pneumocystis is probably not that rare, but people just don't get sick. But if your immune system is sufficiently suppressed, you get sick. Okay, so then this eventually led to a Nobel Prize for the discovery of HIV. And this is kind of funny. Again, these two French scientists, they got it because they discovered the virus. Harald zur Hausen here, who used to do the experiments at the bench I did the experiments as a PhD student, is a little faded here, not because he doesn't deserve the prize, but he got the prize for discovering that human papillomavirus causes cervical cancer and shared it with the other two. It has nothing to do with HIV. This person, black and white photo, was not honored, although he thought for more than a decade that he should be. And as it later turned out, he stole the virus from the French people and passed it on as his own. So this went well for a few years, but with the advent of massive sequencing, people can, reconstruct the evolution of that particular virus and who got it first and, where it acquired its first mutation of the human body. In the end, so he was passing on the French virus of his own. So he was, in a rare case of justice, not awarded the Nobel Prize, although everyone thought he would be. Okay, here are the key publications, if people are interested, but this is not subject of assessment. Okay, now we come to the geographic origin of HIV. And this is clearly sub-Saharan tropical Africa. And what happened here is, it allows me to introduce the case zoonosis, Z-O-O-N-O-S-I-S, and that is when a virus spreads from animals to people. And like the SARS coronavirus. So in this case, the simian immunodeficiency virus turned into the human immunodeficiency virus because the virus changed its host. And as it turned out, strangely, this happened more than once. So what we call HIV is actually, should be called HIV-1 and HIV-2. They are somewhat different. And they also were introduced into the human population independently. It's not once and then they became 1 and 2. No. They were different introduction events. Okay, so that's where it came from. And it doesn't come from a lab or anything. It doesn't come from vaccines or whatever some people may want you to believe. So it came from a monkey. Okay, so the retrovirus multiplication. Here we have an overview, and I think I have little enlargements. Yes, so entry and uncoating. This is very similar to what I told you before, but now it is specific for HIV. So what we see here is the HIV particle, and you see now it's an envelope particle. And I already told you that the genome consists of 2 identical strands. I don't know why they are missing, strands of RNA. So it's a single-stranded RNA virus because the virus particle has single-stranded RNA, but 2 copies of it. So on what you see here is reverse transcriptase. That's the virus polymerase. That is the famous enzyme that can do this unusual copying from RNA into DNA, as we will see later. So, and that makes specific contact to some cells in your body, if you're unfortunate, and these cells have to have the CD4 protein sticking out of them. So that is used to make contact. So then, the viral RNAs, 2 of them, just one shown, gets into the cytoplasm, and the reverse transcriptase, the textbook is not always good. It's not like different and isolated from the RNA, it actually sits on the viral RNA. And you can imagine if these things get detached, they will never find each other again. So therefore, the reverse transcriptase really holds on to its copy, because if that lets go, then goodbye. Okay, so then the RNA is reverse transcribed into DNA. And then we have double stranded DNA in the end, and that one is then moved into the nucleus, then it integrates in a semi-random process into one of our chromosomes, and now it is well hidden. And that is very stable. It will not get out of there in a hurry. So, and then the virus can utilize just ordinary stuff that's already in the nucleus to make RNAs, and then they are translated into proteins. If you make full-length RNAs, you can also make new viral genomes. And this all accumulates in the cytoplasm, and that is then used to form new virus particles that bud from the plasma membrane and therefore acquire an envelope, and then you have a new virus particle. So here we see a virus particle on the right as an electron micrograph, and to the left schematically. So, and you know already a lot of what's going on here, because these blue bits here are your identical copies of the genome, so single-stranded RNA. You see the reverse transcriptase here holding on to its copy of the genome. And then you see a protein that's called integrase, and you see a protein that's called protease. And the integrase is important because it helps the virus to integrate its DNA into the chromosome of the host cell. And the protease is important because there has to be some cleavage done to our proteins in the process. So, and then we see 2 protein shells, like one is the capsid protein, and another shell on top of that is made by the matrix protein. And then here in purple, we have the envelope, and here we have the spike proteins. And in this case, you need to know what proteins make up the spike. And It originally starts as a GP160, glycoprotein 160, that is cleaved into a GP120 and GP41. But they still stay together, and 3 of them make together one spike. And the GP120 part binds to the CD4 protein, and that gets the virus into new host cells. We see this here. So here we have the host cell. Here we have the virus particle. So the virus particle is now drawn a bit more schematically. But you see here the interaction of the virus spike with the CD4 protein. But this is not the only interaction. So at the same time, it also needs to interact either with something that is stupidly called, don't blame me, CXCR4 or CCR5. And so one is the main receptor, that's the CD4, and the other one is the co-receptor. And the virus is a bit flexible, it wants one of those, but it doesn't really care which one it is. Later I will tell you why it is actually quite important to know about these co-receptors. So then capsid gets inside the cytoplasm, single-stranded RNA is released, and then we have that famous reverse transcription where the RNA is transcribed into DNA, and for a moment you have a hybrid. You have basically a molecule that consists of RNA and DNA, and then you degrade the RNA, make another complementary DNA strand, and then you have proper double-stranded DNA that's then moved into the nucleus, and it's integrated into a host chromosome, and then the whole thing is called a provirus. Is there any questions of this? Long terminal repeat that helps to keep the integrity of the ends and also directs in integration. Okay, and then I told you about this strange co-receptor. People think, oh, I don't really care about the co-receptor. CD4 is what matters. But then it turned out that people who have, and this is something you may want to remember for future uses in other units. So this acronym here means we're talking about the CCR5 protein, and this little triangle here is a Greek symbol for delta. Delta 32 means 32 amino acids are missing from the CCR5 protein. So people who cannot make the full length co-receptor but have a little mutation that, got rid of 32 amino acids, are completely resistant to HIV. They will not get infected by HIV if they are homozygous for that mutation. So which in theory is good news. However, you shouldn't bank on it because it's a very rare mutation. So that anyone here has it, the chances are very, very low. So, however, you can see in Southern Africa that the number of people with that mutation is on the rise. So you can see, if you ever had any doubts whether evolution works or not, you can see it in action. So another thing is, if you have a mutation that basically damages a protein everyone happily has, then there's probably a downside to it. Yes, and it is, because it turns out that the carriers are more resistant to HIV, but they are also more susceptible to other viruses. So maybe in the long run, it's not even good if mankind loses CCR5 genes altogether. Okay. So here we have now a more complex depiction of what I just told you, and now this should be, easy to understand. I show it now because maybe it's a bit easier if it's introduced step by step. We have again the attachment, then the receptor mediated endocytosis, that's now outlined here quite nicely, then the release of the RNA into the cytoplasm, then the reverse transcription, the formation of double-stranded DNA, the transport to the nucleus goes nicely through the pores, the integration into the chromosome, you now see a bit more of the chromosome here, with the help of the enzymes called integrase. And here we see that you now have the production of mRNAs. They go back in the cytoplasm where the translation occurs. Then you have the proteins made, and then you have new HIV particles. Okay, this is something that you should be horrified of. And the virus, HIV, is also called a complex retrovirus, and it's not called complex for nothing. And this is a genome map. So it's terribly complicated. And I will spare you this if I ever have a chance to do like a lecture just on retroviruses, or I will happily go through all the details here. But for now, I just like to point out why it is complex, but then you're free to forget it. So it's complex because in different color, you see different proteins that are made out of this nearly 10 kb, 10,000 base pair genome. And you see that some proteins are spliced and some proteins are coded in different open reading frames in the same mRNA. So that could be that a nucleotide in the mRNA is used for two different proteins in a different manner. It codes for different amino acids depending on where you start. So this thing has basically overlapping information. And in some instances, I think in this one is even, and this one here as well, one protein can be used in all of the three possible open reading frames. So the information is very dense in this virus. That's why it's called a complex retrovirus, but if you're happy with it, I will leave it as such. Or do I have demands to go in detail and assess it, then we can do this, of course. Okay, so here we have again our genes, the proteins made, and the function of the proteins. And again, you do not need to learn all of them, but what you really need to know that this virus has a polymerase, as almost all viruses have, and that the protein is of a kind that it can do reverse transcription. And I also told you that we have an integrase and we have a polymerase, and the polymerase is needed because the virus can catalyze some of the cleavaging that needs to be done to form all the individual functional proteins out of a precursor. It can do it itself, but that's where we leave it. Okay, this, however, you need to learn. And that is the progression of an HIV infection if left untreated. And this should actually scare you, and I try to do this a little bit because people should A, seek treatment if they're infected, and B, propagate the knowledge that people infected with HIV should seek treatment. And this is a complex thing, and it's not for nothing here, because I think it is important. So, and we have here different curves. If we go through them one by one. So what we have here in red is the number of virus particles. What we have here at the bottom is the time. And here in red belongs to this axis. So it says virus load in blood. And this is measured as RNA copies per milliliter plasma. It's not infectivity, it's just how many copies you have of that virus gene, because you can measure it so nicely with quantitative PCR. So before you got infected, it is 0. That's obvious. So then you get infected, and you get an exponential growth of the virus to very high copy numbers. But then something happens, and the virus titer, the number of viruses in your blood, drops. Unfortunately, not back to 0. And then you have a period that can last years, where it goes up and down and up and down and up and down, but you see there's a tendency that it goes up. And then you reach a critical point from which onwards it goes up more quickly. And that is where your immune system collapses and the virus gains again the ability to multiply almost in an unchecked way, and then it multiplies until you die. So this is the curve of the virus number measured by proxy, RNA copies per milliliter plasma, in a person not treated but infected. So in blue, we have now the CD4 T cell count in the blood. So I told you as a little bit of an introduction that you have two different T cells, the cytotoxic T cells and the T helper cells. And the CD4 positive T cells are your T helper cells. So they are there because they help the cytotoxic T cells to do their job, and they help the B cells to become plasma cells and to produce antibodies. So they are really important. So, and you have a certain number of them. And then you live quite happily. So, and you see that the multiplication of the HIV virus goes together with the drop of your CD4 positive T cell numbers. And then the virus goes down and the CD4 positive T cells recover. However, the virus doesn't go down to 0, and the CD4 positive T cells do not recover to the level before the infection. And then you have these years where the virus slowly grows and your CD4 T cells slowly deteriorate in number. And if they reach a certain point, around 200 per microliter blood, then everything breaks down. Then you go beyond the CD4 T cell number you have to mount efficient adaptive immune responses. So the immune system collapses, virus goes out of control, people develop AIDS, suffer terribly, and die. So, and this is what happens in untreated people. And then we have a little extra information on top here. So in yellow, acute phase, because of that virus information, you have symptoms here. People feel sick in the first couple of weeks of the infection. Then we have this area in the middle where people feel quite all right, but they're not, because the immune system slowly collapses. And then in the end, you have what's called a syndrome, because it's different from person to person. It depends on, normally the HIV virus kills only a fraction of these people outright. The others die because they cannot control other infections, opportunistic infections. So, and all of this makes this particular syndrome, and then of course you have the horrible signs and symptoms.
What are some of the symptoms of HIV infection?
Yeah, so the early phase, it's like influenza virus infection. You have a headache, you have fever, you feel a bit down, you produce a lot of interferons and the side effects of interferon, for example. And then in the end, it's, you know, we'll talk about this now. You get like, suffer a lot of bacterial and viruses you already had. You never knew that you had them because it was all under wraps and now kind of completely unfolds. Okay, critical number here. 200, and if it drops below, in most people, immune system collapses. Okay, HIV serology. So this is what people measure, diagnose in the clinic. So this is a timeline again. And here on this axis, we have the relative concentration of whatever we are measuring. And after the infection, we have a rise of HIV antigen. So antigen stands most of the time for protein. So there's protein about in the blood, but that goes down because our immune system initially controls the virus quite efficiently. What the immune system cannot get rid of is the virus that integrates itself into the genome. And if the virus doesn't do much in terms of multiplying, it's basically undetectable. So therefore, our immune system fails to kill the very last virus in your body, to eradicate the virus, and that is part of the problem. So, and what you see here in purple and blue, AB stands for antibody. So the body produces antibodies quite quickly after the infection, and they help to keep the virus at check. But only for so long, and then your immune system collapses and the virus is able to produce virus particles again that are not neutralized, because you also lose the ability to make antibodies, and then, you know, people develop AIDS and then they die. Okay, so for controlling virus infections, the T-cell arm might even be more important than the B-cell arm. And because the virus uses CD4 to get into host cells, therefore these T helper cells, who are all CD4 positive, are the main target for the virus to multiply in. So it basically singles out a critical part of the adaptive immune response, therefore in the long run wrecking the immune system. So the consequence are opportunistic infections. And this is a list of infections that normally do not cause dramatic consequences, but in people that are severely immune depressed, and AIDS is one reason why you could be immune depressed, these can kill you. So for example, Toxoplasma gondii. So this is normally something that gets transmitted back and forth between mice and rats and cats. So it's an apocomplexan protozoan. And if you have cats, you have a chance of becoming infected with Toxoplasma gondii, in particular if you cuddle your cats too much. So some people say that this actually affects human behavior. Some people say it affects the mouse behavior. It actually makes the mouse quite, they do not fear the cats that much anymore, which is not boding well for a particular mouse. They also say that it may cause certain changes to the mind of people, but you know, this is thin ice, so we leave it as such. But normally, effects are not very dramatic. However, they are dramatic if you are HIV infected and cannot control it anymore, then you may die from it. Then we see what I circled here under viruses: cytomegalovirus, herpes simplex virus, varicella zoster virus. You probably have heard of those before. They're all members of the herpes virus family. And you most likely have received, apart from two, the varicella zoster virus vaccine. And you too may think of getting them not to prevent an infection, but to prevent the development of shingles later. So I got my first before the benefit scheme pays for it. Because if I tell you what shingles can do to you, you're happily paying the $250 for the vaccine. So, right. So these ones, and many of you will have at least one herpes simplex virus. They can be a nuisance, but they're not going to kill you. However, if your immune system is down, you may have more generalized herpes simplex virus infections that can be much more pleasant and can even kill you. So here, for example, is what people who are immune competent do not really develop to such a degree. So this is a chronic herpes simplex virus type 1 lesion on the lips, and it's also a lot bigger than whatever you may have experienced before. And this is shingles, so it's a segmental infection of the epithelial cells. This is extremely painful, and this is also phototaken from a person with AIDS. And CMV, normally people do not even know they had it or have it, but in immune-suppressed people, it can destroy the retina, so people get blind. So then there are also certain bacterial infections. For example, tuberculosis is a chronic infection that is normally not causing too many problems. However, it can severely impact lung function when it gets out of control, and it has a high chance of getting out of control if your immune system is suppressed. Then we have our pneumocystis. It's now called Jirovecii, so they renamed the thing. And that is causing life-threatening pneumonia. The disease is super rare, and the occurrence of multiple cases of that in California last century led to the discovery of HIV. And here we see an X-ray of a patient with that pneumonia. Okay, Candida albicans, many people know it because they had a Candida infection. Particular women can have Candida infections that come with clinical signs. You can also sometimes see it with babies before they have their normal body fauna and flora. They are a bit more vulnerable to this, but it's all transient. It's normally not that much of a problem, but it can be much, much more pronounced if your immune system is bad. And we see here typical Candida manifestation in the soft tissue in your throat. Okay, and then we have the possibility that HIV/AIDS causes cancer. And this is a bit weird because we have here one cancer that costs a lot of lives because it's not rare in AIDS people. And that's a cancer of skin and blood vessels. And this is not a cancer that is directly caused by HIV. This weirdly is caused by human herpes virus 8, which is normally well controlled, but as with all the other herpes viruses here, is not well controlled if you have AIDS, and then the human herpes virus type 8 causes cancer. And here we have images of Kaposi sarcoma in people with AIDS, and you see it will spread everywhere and is one of the big killers. Okay, the good thing is that we have now a treatment, and the treatment is sometimes referred to as HAART, H-A-A-R-T, highly active antiretroviral therapy, and the trick was to combine typically 3 or 4 antiretroviral drugs, not just one, but a combination of drugs. And there are about 5 approved drug classes that target different things in the virus life cycle. And you can appreciate this now because we spent some time going through the life cycle. So there are drugs that affect the reverse transcriptase. So that's the virus polymerase. That's the one that does RNA to DNA. And we have 2 classes, called sometimes nuke and non-nukes, that depends on whether they try to look like a nucleotide, so that's the building block for nucleic acids, or whether they bind to the polymerase but not in the active center. That's the difference between the 2. And then we have a protease inhibitor, and I told you that the virus has a protease to cleave its proteins that are sometimes made as large proteins, and then they're processed, and then the products of that cleaving step, they do all the different functions, so they have to have an effective protease, which is then another Achilles heel. And we have entry fusion inhibitors. So you know now what happens if a virus makes contact with a host cell and wants to get into this host cell. So there has to be binding established between the virus GP120, part of the spike, and the CD4. And if you stop this from happening, then you will stop the infection. And we have integrase inhibitors that block that little enzyme that takes the virus DNA transcript after it entered the nucleus and puts it into a chromosome. And if you block this, then this is not happening. It's also the end of the virus replication. Okay, so you combine 3 or 4 drugs to get a heart therapy. And heart is good because it usually restores the CD4 T cell numbers in patients. But, that's very important, heart does not lead to virus eradication. So you have to keep treating. So it reduces the virus to replicate efficiency, but it will not kill the very last virus particle or eradicate the very last bit of virus information from your host cell.
Why can't it eradicate that last bit of virus? Like, why can't it?
Because if the virus is inside a host cell, stably integrated into a chromosome, but doesn't do anything or doesn't do much, there's nothing to inhibit. So therefore, every now and then, one of these silent viruses, if you like, gets activated and then has another round of replication. But you may then catch most of the newly produced virus products, but not all of them. And then you have another host cell with an integrated genome. And this obviously is efficient enough to keep the virus in the body. Okay, so therefore, treatment breaks are a rather stupid idea, and you know this. It's the same for taking antibiotics. You get an antibiotic prescribed from your doctor, and you take it exactly as instructed, and there will be no breaks for 3 days because you don't feel like it, so this is a bad idea. However, unfortunately, patients will experience a variety of side effects, and much research in recent years has been undertaken to keep the effectivity but reduce the side effects, and I believe that the 2nd and 3rd generation drugs are now much better. Previously, you could spot the people on the street who have drugs because they have a certain look at it. This is no longer possible. So I think it's getting better, but one thing remains: you will not eradicate the virus. Also, you may question, how hard does the industry try? So, I don't know. Okay, we're not going through lots and lots of different antivirals, but let's do this one. This was one of the first, and it's fairly simple, and maybe you want to take this as the one to learn. So it often comes under the name AZT, and it's a thymidine analog. So here we have the nucleoside, and here we have the analog. So it looks almost the same, but for this bit here, which is different from this one here. And because this is exactly the thing where the chain grows, and you have this group here, then you cannot add the next nucleotide. And so therefore, the nucleic acid polymerization stops, and that's what's called a chain termination. So that will be the end of the replication of this virus. However, AZT must be activated. So you get this one, which is a pro-drug, and you need kinases to add phosphate groups, and then, phosphate groups here. And then this one is the final drug that can be used to be added to a growing nucleic acid chain, and that causes a stop. Okay, here we have a couple of approved drugs. Don't bother learning them, they change all the time. But what you should remember is that they target different things I just mentioned, and that you want to use them in combination, and that they do not eradicate the virus. These are the really critical take-home messages here. These are vaccine candidates. Again, you do not need to learn any of those because they all do not work. So up to now, and I just looked it up before the lecture, there's still no approved vaccination, and it may still take some time until we get one. So, which would be a game changer. So until this happens, we all rely on the antiviral therapy. However, it is possible to prevent infection by treating people even before they got infected, because if you give people the antiviral drugs in anticipation of an infection, then they will never get infected, or if you treat immediately after exposure, then you may stop the virus getting permanently settled in your CD-positive T-cell population. And once that happens, you will never get them out. But you can stop that. So let's say you did something really stupid, you had unprotected sex on Friday night, then within a couple of hours you have a window where you can receive antiviral treatment, and then you will not get HIV infected. However, it's a very small time window. So what also happens is one should advocate for needle exchange programs, and whatever you and I think about morals here is not really important, because if you do not do this, you will just promote the spread of HIV in prisons amongst intravenous drug users, and it doesn't really matter whether they do legal or illegal things. You will just promote the spread of these diseases, which eventually come to people who not engage in these activities. So I think it should be a no-brainer. Then I would like to talk about PrEP and PEP in the last 5 minutes. And PrEP stands for pre-exposure prophylaxis, as mentioned, and PEP stands for post-exposure prophylaxis. So post-exposure prophylaxis is probably the most relevant here. So that is after HIV exposure, but it should be taken within 72 hours, ideally within 24 hours, not 72 hours, after possible exposure. And this is for people who had unprotected sex or had a needle stick injury. Just imagine you're working on one of these emergency vehicles, and you're a nurse, and you can imagine on Friday night there's all kinds of drama and people kind of moving in all kinds of unpredictable ways, and then more often than not, the staff on these vehicles get needle stick injuries, and then there's a chance that there was blood from whoever they just picked up from the street on that needle and then gets into the bloodstream of the nurse or the doctor. So then they could be potentially exposed, and this is a classic case for post-exposure prophylaxis. Okay, so there are these things for Canberra, and I will not read all this. This goes online and you can look this up. The good thing is that if you think you have been exposed and you start your treatment, as I said, within the initial 24 hours, you have a near 100% chance of not becoming permanently infected. So very important. Okay, some more information and learning outcomes, and that concludes it for today. Any questions? Easy, you have 15 minutes. Yes, but I think even if you're not a keen microbiologist and you never want to be a virologist, people should know about HIV because, you know, it's around and it will not disappear in a hurry, and it may save your lives or others.
Would you need something like CRISPR to get rid of it permanently, like a gene
Yeah, I'm sure people think about it, but initially when CRISPR came about and people got the Nobel Prize, and it's a great tool for doing experiments to manipulate the insects and manipulate your laboratory mice and all this, people got really excited. But then CRISPR does not always do exactly what you want it to do. So everyone who has used it in the lab can tell you, yes, in theory it looks all like super predictable and clean, but practically speaking it's not. And this is one reason why fiddling around with the human genome may do more harm than good. So, and then you don't want to be sued because you may have healed someone from HIV, but who unfortunately is now dying of cancer. So I think this is one reason. In theory, yes, maybe earlier or later this will actually be used, and this would be one way of doing it. But then the whole technology has to be developed to be kind of full and idiot proof and not having, undesired effect in every 100th person or so. So this shouldn't be happening.
It's a bit like the telomeres lengthening those is risky because the shortening of the telomeres is like some kind of mechanism of cellular protection.
Yeah. That's true. Okay, then I see most, if not all of you, very soon in the practicals.
Thanks, Mike.
You're welcome.